into a lot of detail on viruses. So I did not include lesions or pharmacology in this lecture, but I think we have time because there may or may not be as much time to spend on the practical part, which was going to be the ImageJ demo. So just for your information. So it's 10:35 now. All of the registered people are here, so I'll go ahead and get started. So today we're going to talk about anatomical tracing techniques. And of course, we are in the Dominic P. Purpura Department of Neuroscience, and he was quite famous for making these drawings, in particular of patients, children especially, with neuropsychiatric disorders. And so we have a lot of these images around the department. So, you know, if we're ever back in Kennedy at some point, you feel free to look at some of the images that we have. So I wanted to give a nod to Dr. Purpura. And so to continue, just to sort of start you off with a little humor here, theory is when you know why something should work, but nothing does work. Practice is when you know that things work, but you don't necessarily know why. And there's a joke that in our lab, we combine theory and practice. Nothing works and we don't know why. So of course, the idea behind this course is to sort of give you some of these insights into why things should work and how they work in the lab. So some of the reasons why we might be interested in doing anatomy is to sort of define cell types. So what are some of the ways we might define a cell type? So there are a lot of different definitions that you could apply, but here a recent review from Josh Sains suggested that, of course, morphology is a big one, and this plays right into anatomy and tracing techniques. So a lot of these cell types have very specific morphology and can be identified by eye. Another one is physiological signatures. So of course, if you're making recordings and you don't necessarily see the cell morphology, sometimes they can actually be identified by their physiological signature. And of course, the past lectures from Luke and Tiago have really focused on physiology as well as in vivo imaging of firing patterns. And so this is sort of a big part of what we've talked about already. And then another way that people are sort of being able to kind of combine morphological and physiological components is looking at molecular signatures. And so there are certain genes that are expressed in certain cell types. And these can sort of lead to confusing definitions or like there are subtypes of types of neurons that are of a morphological class, but within those morphologically similar neurons, they're molecularly distinct neurons. So we might get into some of the sequencing that goes behind defining cells by molecular signature in a couple of lectures. But just to kind of give you an overview of sort of what are the things you might use to classify neurons. And so for this first lecture, we're going to be talking mostly about anatomy and tracing and visualizing neurons. And so, of course, this is a very famous example that was that we discovered the existence of a synapse. So at the time, in between the early turn of the century, the turn of the 20th century, there were two competing theories. One was that there were neurons that were distinct cells that were not connected. The other was the reticular theory that there were that all the cells in the brain were connected. And so by using Golgi stain, so this was actually one of the competing scientists that were in favor of the reticular theory was Camillo Golgi. And so he had developed this stain, and Santiago Ramón y Cajal made a lot of these drawings of neurons in all kinds of parts of the brain, and including from the brain through the spinal cord into the body. There was a really excellent exhibit on tracing that was recently in New York from some of Ramón y Cajal's drawings. But he was able to discover the space between neurons by improving on this Golgi stain. So it just really shows right from the very beginning when stains were first emerging, that they were already being useful for uncovering basic principles in neuroscience. So one of the more modern ideas is to look at connectomics of neurons. And so everyone loves omics, so now we have connectomics, and this can go all the way, of course, from the nanoscale, from electron microscopy. So here's a recent example from Jeff Littman's lab, where they've done dense reconstruction of several neurons using EM. This is obviously a very data dense type of tracing. They have to take these very thin sections, serial sections on EMs. They have specialized slicer that collects all of the sections on a tape. It's not even done by a person. And then of course they have to take all of these images and save it all, and then it gets somewhat automatically but also manually traced. So at the time that this paper came out, this was partially manual. So they just had a bunch of Harvard undergraduates tracing neurons all day. So obviously, I read a review that said that basically if we wanted the entire human brain to be mapped using connectomics, that as of the last decade, we would take up half of the entire storage capacity of the entire world's computing system. Hopefully we've got more computing power now, and I think they are working on ways to make this connectomics EM tracing less dense. But just to give you a sense of how much data that would be. So then of course there's more at the mesoscale, looking at, here we're showing, this is storm imaging of synapses. So of course the presynaptic and postsynaptic sides of a synapse. Here, actually, this is from Perry Krishan in our department. They showed, a whole neuron and some of the synapses along a neuron. So this is, again, at the mesoscale, looking instead of at single synapses, at multiple synapses. And then here's an example, Brainbow, looking at many neurons. So we might talk about how Brainbow actually works in terms of the random expression of different fluorophores using Cre recombinase, but basically looking at many neurons at the same time and being able to trace, using these different colors, trace multiple fiber tracks at the same time. And then here is actually an entire brain. This particular paper, this is rabies stain. So it's basically the presynaptic inputs. I believe this is to TH positive cells in the VTA. And they did light sheet imaging and were able to map inputs across the entire brain. So of course, this is getting more towards the macro scale. And of course, some of the physiology techniques that we've talked about are at the macro scale as well. Hundreds of neurons. Okay, so today we're going to go through tracing techniques. We'll start out with stains and dyes, as we've already started to talk about. A little bit on antibody labeling, which I actually didn't intend to spend much time on, but I realized there were some, you may be using antibody labeling in conjunction with some of your other techniques like viruses and stains. So I ended up spending more time than I thought I would on that. And then the principles behind viral labeling and why we might use it specifically in neuroscience. So we've already talked about identification of the synapse, but I wanted to point out a couple of other somewhat famous examples of anatomy and what they've taught us in terms of principles in terms of brain. And so here, this is Peter Mumbarts and Richard Axel from 1996. They were able to trace several different olfactory neurons. So these, this was a transgenic line that expressed beta-gal in P2A, I believe, receptor olfactory neurons. So here in A and B, C and D, you can see that throughout, this is the olfactory epithelium. So you can see there's quite dense labeling through here. But then when you actually look at the olfactory bulb where those projections are in the brain, you can see they all converge on a single glomerulus. So this was quite a famous example of how we can use tracing to understand how the brain is doing computations, in this case in olfactory system. And then here, of course, Christoph Koch, this was much more recent, a couple of years ago, traced 3 colostrum neurons, and this single neuron originating in the colostrum seems to sort of, I think that the popular press said something like a crown of thorns in the brain, and then there was something comparing Christoph Koch to Jesus Christ, which I thought was a little weird, but it's one of the more recent examples of how tracing can lend itself to sort of making these theories about the brain. And in this case, they're saying this is the brain, this is the seat of consciousness. So basically, this guy is saying this is evidence that this is where consciousness is because these neurons are projecting all over the brain. Take that as you will. So I wasn't actually going to spend time on Golgi staining, but I came across a recent application where they're using Golgi staining in cleared tissue. So I thought that was pretty cool. So again, we already mentioned Camillo Golgi, contemporaneous with Ramon Cajal. And this is basically a silver nitrate impregnation. And it's not entirely clear how the silver nitrate is impregnating particular cells, but it leads to really sparse staining. And so that's really important when you want to look at morphology of neurons, that you don't have dense staining, because then you wouldn't be able to see individual projections. So of course, this has been used not only to look at the entire neuron, but more recently look at these spines in different preparations. In this case, this is a MEF2C knockout. So you can see here, they've looked at quantified the spines and control and knockout animals, and there's more spines in the knockout animal. And then here they've done some improvements. So again, as I said, Ramon Cajal himself had improved on the original Golgi stain. Most people now are using something called Golgi Cox. And then, so it's been combined with cubic now, and cubic's one of the quick ways to make the brain transparent. And so now you can sort of do whole brain imaging if you need to do that for some reason with Golgi Cox instead of something else. But the advantage of Golgi is that you don't need to know the identity of the cells. You can, this will sort of give you, as you can see, a salt and pepper impregnation across brain structures. And it leads to sparse staining, so you can really see all of the processes. So now we're going to move into dyes, dye tracing. And so this is anterograde versus retrograde. So when we're talking about neurons, we're talking about, are you putting the dye into the terminals, or are you putting it in the cell body, and where does it get trafficked? So in the case of retrograde, the dye is being put at the terminals, and it will be taken up into cell bodies. So it travels up the axon retrogradely. And in the case of anterograde tracers, they seem to prefer to go in on the dendritic or cell body side and then go out to the projection. So that's the difference between retrograde and anterograde. And in this case, for dyes, we're not really talking about transsynaptic labeling. There's some idea that some of them could be transsynaptic, but for the most part, they're really staining just the cell that they're touching. They're not crossing synapses. Transsynaptic stuff we'll talk about more in terms of viruses. So there's a good review from 2015 covering all of the different types of dyes and stains that are out there. I'll talk a little bit about HRP is one of the oldest horseradish peroxidase, and it just gets taken up into cells by endocytosis, so it really doesn't traffic into the cells very far. So it's not exactly a retrograde or anterograde. It really only stains at the bouton. And then of course you have molecules, fast blue, Florigold is one that's quite extensively used even now. Then there are biotinylated dextrin amines, plant lectins, which there are some that are anterograde and retrograde, retro beads, which are microspheres, Some of the toxins, especially cholera toxin beta, prefers to go retrograde. And then these, I'm not as familiar with the biosite, and of course is used a lot in physiology if you want to fill a cell. And then di and dio are just di. They go both anterograde and retrograde. So if you're just looking to see where you dropped a pipette, for example, in the brain, you might use di or dio, but it's going to go both directions. Some of these others have more preference for one direction or the other. And why do they have preference for the direction? That is dependent on how the dye or the protein enters the cell. So of course, as I said, HRP is really passive. So some people have used HRP quite successfully if they're just looking at vesicle transport, so they can see vesicle fusion, because the HRP will be taken up, and they can of course bias towards more endocytosis if they treat the cells with potassium. So you can see there's a lot of intake of HRP. So this method is still used now for that, but it's not really used for tracing. Dextrin amines seem to enter the cell at an injury site. And then CTBs enter by binding to GM1 ganglioside. And it appears that this is mostly axonally localized. I couldn't find a reference that definitively said that, but because CTBs primarily are retrograde, it appears that that's primarily where they are. And then WGA and PHAL, so two of these plant lectins, they bind to sialic acid and N-acetylglucosamine on the membrane. And so that allows them to be taken into the cell. Retrobeads appear to be endocytosed, and then Florigold can either enter through an injury site, or most people prefer to inject Florigold via iontophoresis. So here's an example. So you can see that so basically iontophoresis is putting an electrode into the pipette and then giving a pulse, and then it allows the lipophilic dye to sort of stay locally and enter cells in the local area. So here you can see that if you inject into the medial amygdala, you can get retrograde transport to the presynaptic accessory olfactory bulb cells. So the reason I put this on here is that this gives you kind of a framework for why something might be anterograde or retrograde. And so we're going to talk about that too in terms of the viruses, why they go certain directions. And as long as you know that there's something you can look up, you can basically, anything that you're working with, you can look up, see how it enters the cell, and you can figure out whether it should be anterograde or retrograde. So what are some of the applications for tracing? So in this case, this is WGA conjugated fluorophores that have been used for anterograde tracing. So here you can see the barrel cortex, and basically they've gone in and put the different WGAs in neighboring barrels, and then you can see in the thalamus where they project that there's sort of this topography that matches the barrel. So this is kind of an aesthetically pleasing example of WGA anterograde tracing. We tried this, I tried this in the lab. It was not as easy as they made it seem, but this was a nice example, 2015. And then of course, CTBs for retrograde tracing. So here you can, if you want to determine if your cells are, for example, bifurcating a projection, you can put dyes into a couple different, or even 3 if you have enough spectral separation in your imaging and how many sites you need to inject. But in this case, we have genetically identified neurons labeled with BFP, and then put far red and 48 in different projections, and then quantified how much overlap there was. So these are some of the things that you can do with these tracers. So I don't have a lot to say in terms of using them in practice, other than a lot of it has to be empirically determined, unfortunately. So based on how how long the projection is that you're trying to trace, how many you might be trying to trace at the same time. You're gonna have to figure out how long you need to let your dye retrogradely travel. So this is just an example that showed that there is a difference in how many, in this case it's retinal ganglion cells. If they let CTB incubate for a week or 2 weeks, they get a significant difference in how many cells they've retrogradely labeled. In the case of Fluoride Gold, at least in this, this particular projection, they didn't see much difference. But again, this is also projection dependent. So it depends on where you're injecting, how far the site that you're trying to see retrograde or anterograde labeling is. And then again, you can sort of push it in a direction if you just inject more dye, if you want to load it with more, if you're looking for a long range projection, you might want to put more dye so that you see more of it taken up. And then how much volume you put in, that's of course going to change what brain area, how many cells you label on the side of your injection site. So that's obviously going to change how much uptake you get. So typically the injections are very, very small. And then because they're dyes, the one other thing I will say is that when you're using, for example, a glass pipette to inject these dyes, you have to be really sure that you don't have precipitation of your dye at the tip of the needle. So you really have to make sure it's clean. If you're doing multiple injections, you probably need to clean the pipette tip or potentially just cut it a little bit so that the precipitate's not sitting there. There was one other thing, but I forgot what it was.
Anita?
Yes.
How toxic are these dyes?
For the most part, that was one thing I was going to say too, is that I've never really seen people in practice use them for more than a week. In theory, they're not toxic, but you are loading them with a dye. So the toxicity really relates to how long you're trying to inject it for. And that can, if you see toxicity, you can lower the concentration to make sure you're not damaging your cells.
And Anita, I have a question. So I'm a little confused, I think, by the distinction, or if there's a distinction you're making between, say, for example, retrograde and transsynaptic retrograde. You see what I'm saying? So is it just you're talking about being uptaken by terminals, or you're saying being uptaken by terminals, transported to the cell body, and then transsynaptically being uptaken by the next set of terminals? And are all of these, you might have said this and I just missed it earlier, are all of these you're talking about transsynaptically?
The dyes typically are not transsynaptic. They can't butt off of a cell and infect the next cell.
Right. Okay, but then the WGA is transsynaptic though, right?
I think there's some thought that it can be. I think the efficiency is not very good.
Are there any of these that are efficient transsynaptically?
None of the ones that I've seen. So it really, so again, it's about, for the retrograde and the anterograde ones, it's really about hitting that first order cell. And then it either travels to the projections, or if the projections are there, it travels to the cell body. So it's really first order. Yeah, as I said, the WG, there's like low efficiency. So it could be, but to me, the mechanism is not clear, I'll say that, of why it would travel all the way and then pinch off. And then, so presumably if you load it, maybe in a concentration dependent manner, you could get enough loaded that enough of it gets to the, let's say in the case of WGAs, enough of it gets to the axon that it buds off and then can, through vesicular transport, get into the next cell. But it's not entirely clear to me what the mechanism would be for it. Okay. But I think people are trying to use the tropism of it to basically infect it with a virus, and so that basically they can get that mechanism to work. So actually someone I know, Steve Santoro, was trying to use wheat germagglutinin to basically virally express something that would then travel anterogradely, but haven't heard any updates on that.
Thanks.
Yeah, so I forgot what the other thing I was going to say about using dyes in practice, but basically, yeah, you really need to make sure that you don't have precipitation, because then your actual dye is not going to be deposited into the brain. So next we'll talk a little bit about immunohistochemistry, and as I said, this is not necessarily widely used for tracing, except for in certain parts of the brain, but you may actually end up using immunohistochemistry. For one thing, a lot of these dyes that we talked about, you don't have to be conjugated to fluorophores. They could be conjugated to HRP or something that you actually label with an antibody. And then of course you can amplify any of these, but I just thought it would be good to go over a little bit, since it's kind of a practical course, some of the things that you may or may not have known when you're trying to use antibodies in the lab. So of course, you probably are familiar with an antibody structure. You have the variable portion here that's actually going to bind to your antigen, and then you have the light chains and then the heavy chains. So this part is constant. This part is going to change depending on what you're, so this part, the constant part, is going to be whatever the host animal is. So that becomes important when you talk about primary and secondary antibodies. So not all antibodies are equivalent. So basically, as we talked about, you have this, these epitopes, and those are little pieces of your actual antigen. And that's what your variable portions are going to recognize. So typically, if you have a polyclonal antibody, that means that they're recognizing different epitopes. So it's a mixture. So how do we make polyclonal antibodies? I'm sure you guys have seen these kinds of diagrams. You basically put the antigen into a rabbit. You get activation of the immune system, and you start getting antibodies against pieces of the antigen. So then you can take those cells and obtain antisera and basically get the antibodies out of the serum. So typically when we talk about polyclonal antibodies, we talk about rabbit, chicken, rats, camelids, sheep, and goats. Monoclonal antibodies more or less come from mouse, and basically they take only spleen cells. So why do they take the spleen cells? Well, that's where a lot of the immune cells are. So basically they can take the spleen cells where they've got a high concentration of the antibodies and basically select them. They make them into hybridomas so that they become immortalized, and then they can separate out actually. So they start out as polyclonal here in the spleen, but then you can basically capture the monoclonal, so meaning that it's just one antibody. So these can be kind of more expensive because you have this step. So you're not just bleeding an animal, you have to take the cell, you have to kill the animal, and then take and make these, screen for these antibodies. So again, here with the polyclonal antibodies, they're going to be recognizing different epitopes, but here with monoclonal, they're going to be against the same epitope. So you're going to have maybe a lower background, a cleaner staining. You might have lower signal. And if you're using a mouse antibody on mouse tissue, sometimes you can run into problems doing that. I just wanted to say a couple words about camelid antibodies, mainly because I needed to show this meme of a llama. But basically they were discovered, I think about 20 years ago. They're single domain antibodies. They only have the heavy chain, VHH. And so this is much different than what you see with here you have the heavy chain and the light chain, but here only the heavy chain exists. And there are actually some diseases in humans where there is heavy chain only. So it took some years to characterize and make sure that these antibodies were actually functional, because in humans that have heavy chain only, they're non-functional antibodies. And so, of course, through the years, they've sort of developed this method of only using fragments of antibodies. So here you can just use this VHH fragment from the heavy chain only antibody. And so that lends itself to some cool applications. Basically, it makes staining a lot easier. And then you can do some cool things where you conjugate it directly to a fluorescent protein. It can make doing immunoprecipitations a lot easier, because you just have to deal with the fragment of the VHH. And then they've talked about applying it to make to facilitate transgenic expression of genes. So basically, if you recall, VHHs are small peptides, so they're just a few, they're pretty small, so you can actually get them to drive expression of genes in cells. So what does a camelid nanobody look like compared to a mouse monoclonal antibody? So here's the monoclonal, the nanobody that's directly, this is the directly conjugated nanobody, and this is the nanobody developed by a secondary. So you can see the staining is very nice. And in this case, this is a synapse, synaptic protein that's stained here. So you can see you get a lot better localization if you're doing something that requires localizing antibodies. So then we have antibodies in practice. So basically, every time you get an antibody, it comes with a data sheet. It's going to tell you if it's monoclonal or polyclonal, what the host is. It's going to tell you how to store it. Some of them can be stored at 4 degrees. Some of them have to be frozen. Basically, this is me telling you, read your data sheet if you get it, because I don't know how many people have told me their antibodies don't work, and they are either using the wrong dilution or they're not amplifying it. I think on here, somewhere, oh yeah, here it is. It says use antigen retrieval for this. So it's right there on the data sheet. So there's no reason to do your staining and then say it doesn't work, and it's because you skipped your antigen retrieval step that was clearly labeled on your packaging. Don't make a meme out of me, though. So this is another thing that I've heard. Can I skip this step with antibody staining? So this is just to show you what the purpose of doing your blocking step. So you typically block with the serum of the animal from your secondary antibody, and that gives you a lot cleaner staining. Antigen retrieval, we just talked about that particular antibody required retrieval. And the reason that you might need to do antigen retrieval is because your epitope may actually be inside of your cell. And in particular, this is a BRDU antibody. So BRDU incorporates into DNA. So it's not only through the cellular membrane, it's through the nuclear membrane, and it's in potentially in chromatin. So there's a couple antigen retrieval steps. Usually you do trypsin as well as a heat antigen retrieval. And so you can see a big difference in not staining and staining for that one. And then if you are doing amplification, which I'm not going to talk too much about today, there are endogenous peroxidases in the brain, which is how the enzyme deposits the color in the amplification step. So if you don't quench endogenous peroxidases in brain tissue, you're going to see background staining. So this is when you don't block the background, and this is what it looks like when you do. So no, don't skip the step. If you got a protocol from somebody, especially me, don't ask me if you can skip the step. And then one final thing that is really important for my lab, at least, is we look at neuropeptides quite a lot. So this is another reason you might say, oh, my antibody's not working. So neuropeptides are trafficked to axons. They're trafficked into vesicles. So we cannot see cell bodies. So there may be a reason based on cellular localization why you might not see your antibody. So we actually use colchicine. Colchicine is a drug that's used for gout, and I didn't go down the rabbit hole of figuring out why it treats gout, but basically it blocks polymerization of tubulin. And so that way you can get retention of peptides. So this is AVP, AVP with colchicine. AVP always shows up. CRF does not show up, corticotropin releasing factor. This is the paraventricular nucleus, and you can see with colchicine it does. And this is a extremely accurate schematic of how colchicine works. It ties. Nobody's laughing. Okay, it's not a good joke. Anyway, it made my day when I found this illustration of the colchicine tying a knot around the axon. So, does someone have a question?
Yeah,
I was just gonna say that I'm sorry. I did laugh, but my mic was muted. So.
Same.
We at least got one.
Okay, thanks.
I had a question about the concept you were saying. You said it blocks the polymerization, but are these these stains are in tissue that are fixed?
No, the colchicine has to be administered 24 hours before you sacrifice.
Oh, okay, that makes sense. Okay.
Yeah. So it's a pain.
And can you comment on administration and how easy it is to do in the lab actually?
So colchicine can
or
be
not.
So in the case of the PVN, if you're just looking for something close to the ventricle, you can do an ICV injection, which is not so difficult. You really have to be careful with the concentration. It's obviously because it blocks tubulin polymerization, it is pretty toxic. So only 24 to 48 hours. And then you have to be careful with the dosing. I think it's like something like 10 to 100 microgram per microliter, but I don't remember the specifics. So it's not difficult to do, but if your area of interest is far from a ventricle, you may have to do a local microinfusion, which I have seen people do infusions into like hippocampus. Specifically, they were trying to induce toxicity using colchicine, so it wasn't for this purpose for antibody staining. So it might be more of a pain if you have a different brain area that's not close to a ventricle. So then I just wanted to give a couple comments on equipment in the lab. So how to keep tissues on your slide for 2-3 days staining process. And I'll just tell you a little story about my first job in a lab when I was 20 years old in the University of South Carolina. I got to gel sub slides. That was my job. It was terrible. So don't forget that you should just buy charge slides, because gel subbing slides takes you 2 days. You see this? This is after you spend all day making gel, which by the way, gel is not great to work with. It can polymerize in your brain, so you definitely need to be careful working with gelatin. Then you have to use a bunch of it, coat your slides, and then you have to let them dry and then put them back. It's a pain in the butt. Buy charge slides, and don't forget that you have to have the charge slides, otherwise your tissue is going to fall off. And the reason this works, in the case of gel, it's basically a substrate that sticks to your tissue and it's in a thin section. In the case of in this case, these are Superfrost Plus slides, but there are other brands. I'm not paid by Fisher or anybody. The tissue sections carry a slightly negative charge when they're immersed in neutral pH. So having the positively charged slides will help your slides stick on the tissue. So if your tissue's falling off, ask yourself, did I gel sub my slides? Probably not, because only people did that 20 years ago. I should be buying charged slides. And then in terms of mounting media, this can be really important. You have stuff that does not, actually I found a VectaShield that does hard set, which I was really excited about. So if you don't have hard setting mounting media, then you're going to need to put nail polish on your slides to adhere the cover slip, so that when you're imaging your sections, your cover slip doesn't slide off or damage your tissue. And then Prolonged Gold is what Costa likes in our imaging facility. It can make little bubbles, so you got to be careful of that. And then what's nice about your mounting media, a lot of times it contains DAPI. So if you want to see your cell bodies, you don't have to do an extra step at the end of your staining. So here we can take a break. We're going to talk about viruses next, but I'm not going to mention coronavirus at all. So we'll give you guys 5 minutes break, and then we'll come back and talk about viruses.
It's a nice slide.
And I definitely took a lot of time talking about basically things I wish I had known about viruses when I first started using them, and not as much time on the actual tracing that you do with viruses. But of course, Luke and Tiago have talked about fluorophores, GFP, mCherry, GCamp. So you guys have heard about those kinds of things. And if there are specific viruses you guys want, I only had time to go over in depth a couple of them. So if there are ones that you guys want me to go in more depth on, just email me and let me know. Happy to do that. So things that you need to know, basically, there are a couple of reasons why you might use viruses. One is that they're a backbone for delivery of genetic material, and because of how they enter cells. So basically their tropism, and that's through the capsid. So basically these capsid proteins by and large are coming in through either receptor mediated, maybe it's not the best word, but they basically tether to the cell membrane somehow, and can get inside the cell basically through endocytosis. So different serotypes and different viruses have different tropism. And as we'll talk about, some of them actually cross synapses as well. Well, a lot of them, obviously, a lot of them can infect if they are replicative, but a lot of times we remove replication when we're trying to use them in the lab. So we'll talk about how you can basically trick a virus into going a synapse based on its properties. And then basically the viruses that we use, we try to make sure that they don't actually have virulence factors. So we'll talk a little bit about that, because I know there are people who get a little nervous when you talk about using viruses for manipulations and especially for gene transfer. But if you are handling the viruses properly, they are only going to deliver some sort of GFP or something to your body, and they can't replicate. So they're pretty safe, the ones that we're using for neuroscience. So just to kind of give you an overview, I don't know if you guys have been binging on news, maybe you've thought about this more than you want to recently, but basically viruses attach and enter the cells. Some of them integrate into the host genome. That's not necessary. Then they replicate and of course create new viruses that then bud off, they release, and then start the cycle again. So those are wild type viruses. So what do we use in neuroscience research? By and large, people are kind of moving toward adeno-associated viruses, and I'll talk about the reason for that. Lentiviruses, retroviruses, herpes simplex, pseudorabies and pseudotyped rabies, these are not in any way related, and we'll talk about those in detail. Canine adenovirus, so those are the few that I thought of off the top of my head to go through. Maybe you use some other ones, so I'm happy to talk about others. So obviously there are pros and cons to using particular viruses to facilitate gene transfer. So there are some reasons why you might choose one over the other. So here's herpes simplex virus, adeno-associated virus, retrovirus, and lentivirus. So whether or not they integrate into the host genome, as I said, they don't have to. And in fact, HSV is always episomal. AAV has been mutated so that it mostly remains episomal, even though the wild type can integrate. but only when it's in the presence of other factors, which I'll talk about. Retro and lentiviruses, these have been derived from HIV viruses. These integrate because they're retroviruses. The cloning capacity is a big reason why you might choose something. The capacity of HSVs is up to 150 kb. The ones that are replication deficient are 40 kb because they added some things in there to keep it from replicating. The payload for AAVs is 3.5 to 4 kb, so it's a lot smaller. Retro and lentis are a little larger at 7 to 8 each. The tropism for these, HSVs broad, AAVs pretty broad. Retroviruses can only get into dividing cells. Lenti doesn't have a preference. And then how long they express might be a reason why you would choose one or the other. HSV expresses quite rapidly within 12 hours, and it expresses for days. If you have the replication deficient kind, it might be months, but it's still not as long lasting as some of these others. So AAV, they basically advertise that it's 6 months, but I think it's like years also. Retro is a little shorter, primarily because the cells are dividing and the signal is going to get diluted out. And then lenti is years. I couldn't actually find the expression onset for lenti and retro, but I think it's fairly fast, on the order of days. But I think a lot of people are using lenti in cell culture, so I didn't want to post that information. This is all how long it takes to express in vivo. So expression onset for HSVs is within hours. And then for AAVs, it's 7 to 10 days. And I'll talk about why there's a latency there and how they try to get around that. What are the disadvantages? So HSV does cause some immune response, so it can be a little bit toxic to the cells. AAV is not toxic, but it has a very low payload. And there can be some episomal expression. Because these actually do integrate into the genome, they can be mutagenic, both retrovirus and lentivirus. And then for practical reasons, you might want to choose them based on what the BSL level is, what you're cleared for. So HSVs, retros, and lentis are all BSL2s. They have extra precautions in place. AV is BSL1. So one thing you might want to
Anita?
Yes.
Before, tropism means what it's going to target, but it has nothing to do with like the promoter that will be, when you say tropism broad or dividing cells, it's regardless of the type of cells, if it's glia or neuron or whatever.
So these, yeah, so basically here we're talking about the more or less how the capsid is infecting, not anything else that you've engineered into the cell, like the promoter or enhancers or any of that stuff. So basically this is just talking, so broad meaning a lot of, so you can actually go and look up, there are a lot of tables out there that you can look at, but basically it's like what types of cells, so like heart, lung, brain, kidney, and then also dividing, non-dividing. And then with some of these, you can even get into the nitty-gritty of like, is it preferring glia versus neurons? And some of them have biases for it, but we'll talk a little bit more in depth specifically for AAVs. I have some information about that. It was too much to go into detail for all of these, but there are some subtleties, but generally speaking, they can infect, but we'll talk about how they infect and why it might be limited. And that's part of the reason I wanted to give this talk, was to the spirit of like, not necessarily focusing on, oh, this is how you get tomato in a cell and trace, but like, how does the virus actually getting there and why that might affect your experiments or what you see. So I spent more time than I thought I would on some of these just general virus things.
Just one quick comment, one quick comment, Anita.
Yes.
You know, so AAV, so I'm not sure why Rachel Levy put 2.5 to 6 months. I mean, in neurons, it's been shown it's permanent. It's been shown out to like 10 years in primates, but it depends on, so if you have a dividing cell type, then it gets diluted out. So then the duration of expression depends on how rapidly the cell divides. I'm not sure why she put that, but it's in post-mitotic neurons, it's permanent.
Yeah, yeah, and I'll talk about how they've engineered to why it's so stable. But yes, my understanding is that it's always there. So how big is your payload? So this is a truck that's obviously way exceeding its payload. So this is something that's gonna be a practical concern for what you're choosing, HSV versus Linty, blah, blah, blah. So, and this is especially comes into play if you want to use AAVs, because you do have limited space in your AAV backbone. So I just wanted to give some insight into how large some of these constructs can be. So Chandra-Dobson is 1662 base pairs. You know, these are all in that range, Cree recombinase, GCAMP. Most of the fluorescent tags are 700 to 1400. Does anybody remember why this would be the range, exactly double? Does anybody remember? No? Like GFP is 700, another one is exactly, no. So it's because they're dimers, the same gene twice. Oh, everyone's shaking their head now. Nobody wanted to speak up, okay. So then your promoter is actually going to take up some space. And the promoters actually, there's a much larger range of how much genetic space a promoter takes up. So this gets to Noelle's question a little bit. If you're looking through some viral catalogs, you're going to see different promoters. So one that would express anywhere is going to be the chicken beta-actin promoter, CAG. It's pretty small. It doesn't take up a lot of space. You can get much more specific. Synapsin is 485. GFP is much larger than that, 2000. And then the weird thing, I was trying to find CAM kinase 2. I don't know if this is if it's patented or something, but I couldn't find the size of CAM kinase 2 promoter to save my life. But I wanted to tell you about that, but I could not find the information. All of the annotations on AdGene, it's missing. So your promoter is going to take space, and then so this is actually gets to why sometimes we prefer to use a transgenic animal to express Cree, and then to go and put a Cree-dependent gene in something, is because a lot of promoters are not defined, or they're just enormous. So like CAG and synapsin, these are probably what we would term minimal promoters, meaning that we know how much genetic material we need to get promotion, so expression of something downstream. So for example, getting back to neuropeptides, for example, for vasopressin and oxytocin, we know what the minimal promoter are, so you can actually get oxytocin into a virus. It's small, oxytocin promoter, I should say. But some of the peptides that I work on, gallin and urocortin 3, there is no defined promoter. You just call it the 3' side, and you don't know what's up there. So, whoops. So basically, there's a huge chunk of it, and we put that in the transgene. And then you guys have talked a little bit about control and enhancer elements, but again, these are going to take up space. The cleavage site, P2A, iris signal, WPRE, that enhances post-transcriptional expression. Poly A sometimes is good if you want to enhance expression in eukaryotic systems, which obviously most of the time in mice, we're talking about eukaryotic systems. LOXP sites, so all of these are going to take up space. So this may, depending how many things you're trying to shove into your viral backbone, you might need to choose a specific virus. So I'm going to talk a little bit about AAVs and how they've been engineered to do what we want them to do in the brain. So basically, this is the entire AAV. So you can see there's only 2 open reading frames for the rep and cap material. So the first one is rep, and it contains the replication elements. The second is cap. It contains the capsid protein sequences. And then the other thing that's important are these inverted terminal repeat sites. And I'll talk a little bit more about how they're used, but these are basically the obligate genomic material that you need. And the others can be supplied in trans, which we'll talk about what that means. So the area that you can use is 4.8. So the whole thing is a little bigger than that, but this is how much your payload could be. It's single-stranded, but it uses the host DNA to make the complementary strand. And this is necessary for expression. When it does, so the AAV itself is called adeno-associated virus. It was thought to be a contaminating element of the preparation for adenovirus because it actually cannot do anything in the absence of adenovirus. So here I talk about that. The integration requires co-infection with adenovirus, herpes simplex virus, or some sort of cellular stress of genotoxicity like UV radiation or hydroxyurea. And it actually binds in a particular site. So this is part of why it was developed, but in fact, we don't use the integration for neuronal expression, but it does go into a particular site. So retroviruses randomly integrate. AAVs have an AAV site in the human chromosome 19, which binds to rep, and that's how it integrates. So even when it does integrate, typically it's not random. We know where it's going, which can be pretty important when you're talking about expression of stuff in the body. So why did they end up getting developed? I've alluded to this a little bit. They're completely non-pathogenic as far as we can tell, no disease, low or no immune response. So obviously with adenovirus, it was used originally, but adenovirus is not used anymore because it causes a big immune response. And it's non-replicative in the absence of these other factors. And it's biosafety level 1 due to these reasons. So I'm going to talk a little bit about how pseudotyping of AAVs works and what the preferences for different serotypes of the virus are. So basically, all you have to have in your AAV plasmid with your gene of interest is those ITR sites. Everything else is supplied in trans. What does that mean? Not with your gene of interest, basically, and it's not in the genome. So these are supplied in trans as well as a helper plasmid. This contains the HSV or the adenovirus that allows it to replicate, because obviously when you're trying to make the virus, you need lots of copies of it, so it has to be replicating. So you basically have a cell line where these plasmids are supplied in trans, and then your supernatant is going to contain those particles. You purify it, and then you titer it. So titering can be important. We can talk about that. Right now, most of this stuff coming out of AdGene is like 10 to the 13th infectious particles. So it's extremely high titer. Back in the day, it used to be low titers and you would worry about that. Now you have to worry about the titer being too high. And as I mentioned, the ITR is the only element that have to be supplied in cis. And why is that? Well, once you get your virus, you don't want it to replicate. So in fact, you don't need those other things. You don't need more capsid because it's not exiting the cell. You only need it to enter the cell. So as long as it's packaged in the particle with its appropriate capsid, you don't need it to actually be expressed. You just need your gene of interest. So basically you're using it to enter the cell, deposit the payload, and that's all you want it to do. You want it to do absolutely nothing else. So there is a variant called self-complementary AAV. So this gets at why there's a little bit of a latency for expression of AAV. So SC-AAV is for self-complementary AAV. So obviously if it's self-complementary, that means you cut your payload in half because you've actually complemented your sequence. But why would you do that? So basically when your single-stranded AAV enters the cell, it has to go through second strand synthesis using the host genome machinery. So if you have the self-complementation, you can skip that step. And so this is why there's a little bit of a latency, the 7 to 10 days to get expression. And this much shorter, it's on the order of a couple of days for self-complementary AAV, but again, you're gonna have a smaller payload if you use that one. So this might be more getting at what Noelle was asking about, which is the serotype tropism. So I think I might have a separate slide on this, but more or less when we talk about serotypes, we're talking about the capsid proteins, not the genome backbone. So almost always we're using AAV2 as the genome backbone, but because we're supplying the capsid in trans, you can have any capsid that you want. So we're talking specifically about the capsid when we talk about serotype. Because I always get confused when it's like, no, it's AAV 2.1, it's 2.1, it's 2/1. It means the capsid, the second one is the capsid, the first one is the backbone. So again, in the central nervous system, most of the 11 known AAV serotypes are infectious to some degree. We'll talk about the specific kinds of cells. There are some that prefer the heart, only AAV2 in the kidney, 7 through 8 in the liver, some for the lung, pancreas, photoreceptor cells, which of course are peripheral nervous system cells, and then the retinal pigment epithelium, skeletal muscle. So you have different tropism. And then there are some new serotypes that have been engineered. DJ is actually a mixture of like 1, 5, and 9 or something. I forget the exact serotypes, but it's basically a combination of different capsids, so it can have tropism across different cells. DJ8 is just a new version of that. I'll talk about AAV retro, which is supposed to be retrogradely transporting AAV. And this is a little bit misleading. Some AAV9, I think, also has retro or anterograde. Anyway, a lot of them can go retrograde or anterograde based on the original serotype, but this has basically been mutated to prefer retro. And then here again, PHPB was already one that could be taken up from the periphery into the brain or into other tissues as well. And this has basically been engineered now to prefer CNS tropism from peripheral injection. So we'll talk a little bit about that. So one thing that I wanted to talk about was basically how long it takes stuff to infect, and then how stable it is. So again, you know, they're getting expression up to 9 months. As Luke said, it's probably permanent because it exists as an episome. So basically, because the rep and cap have been taken out and expressed in trans, the double-stranded DNA exists as an episome, which means it's outside of the DNA and it will be there forever. If a cell divides, it will not go with the divided cell, but it will stay in a post-mitotic neuron, as Luke said, maybe forever. But I did want to just go through this because I actually, in the neurons that I typically look at, I use serotype 1. So here again is this notation 2/1. That means the backbone is 2 and the serotype is 1. So this actually has pretty high expression from right away, from 4 days to 7 days. There's not a big difference, and then it kind of levels off. Some of them are more slow to progress. So for example, 2.5 has a slow onset. You have a big expression here after 14 days, and you're gonna accumulate signal over time. And then here with 2/8, maybe it's a little more stable. So 2.1 is high from the beginning, but it's pretty stable over time. Here with 2, so this is a percentage from the day 4 in panel B. So you can see the 2/5 is getting this huge increase. So this, for example, if you are trying to express GCAMP or something, you may need to keep something like this in mind, that you may have more cells expressing GCAMP, you know, from day 7 till day 9 months, whatever black is in here. So just keep in mind if you're doing long-term expression studies that this could affect how your signal looks day to day. So here is just a typical map that you would find on Addgene if you're looking. And again, this is the backbone. So again, it's AAV2 ITR. So this is the obligate genomic material that you have to have in your construct, and it comes from AAV2. So that's why you see 2. It's because the backbone is AAV2. So again, transport direction, they do have preferences for anterograde or retrograde. And then here they've actually gone and looked by brain area. And so there are some preferences. So for example, AAV5 here looks like it's very highly expressed in striatum and hippocampus. And this could be due to the cell types that are there. But essentially the reason comes down to what receptors are expressed. So how the serotype gets tethered to the cell and taken in. So we'll talk about how to think about that. So yeah, you have differences in certain brain areas. And then here again, neurons versus astrocytes versus microglia. Most of the AAVs prefer neurons. It looks like some serotypes have tropism in astrocytes and then very few for microglia. So this may be something you want to keep in mind. So you can basically use the capsid as well as a promoter to bias your infections toward a particular cell type. So again, why would a serotype have a preference for a particular type of cell? And it basically has to do with the receptors. So in 2006, it actually wasn't known what the receptor was for 1, 6, 7, and 8. For 2 and 3, it was HSPG. And then co-receptors are basically things that help it tether. So for example, coronavirus does have a co-receptor. I said I wasn't going to mention it and I did it, I'm sorry. And then AAV4 and 5 have these sialic acids. And then for AAV5, it was co-receptor PDGFR. So that was as of 2006. But then one particular receptor that was necessary for all serotypes was identified in 2016. So basically they found across different serotypes that if you had knockout of this particular receptor, that you almost had no AAV infection. And so that was in 2016. So then if they do an adenovirus, it actually doesn't have an effect because adenovirus goes in through a different mechanism, but basically this AAV receptor is necessary across the serotypes. So again, there are other receptors that can help a virus enter a cell. So basically the complement of receptors that are expressed by your cell of interest are going to facilitate entry of AAVs into the cell, but at least it needs to express the AAV receptor. So I think there are some people trying to basically delete the AAV receptor in particular cells and basically do cute experiments where they only infect some neurons but not others using an AAV virus. So there's some little genetic tricks that you can think of using an AAVR knockout in particular cells. And then here is just the crystal structure has been determined of AAV receptor, and they show it bound to viral particles. So basically this idea of there being a particular receptor that allows entry based on the interaction between the capsid proteins and those tethering agents was used to essentially test different capsid proteins and just inject them in known projection areas and just see based on you know, randomization of the capsid protein genes to see what would have tropism for retrograde transport. And so essentially this paper from Janelia researchers in 2016, Alla Karpova and Lauren Luger, They basically developed a bunch of different AAV2s. Again, they're based on the AAV2 backbone. The capsid has just basically been mutated until it has preferential uptake. So basically, I have tried retros, and the retro works, but a Cre-dependent retro does not work, and we can talk about that more in some other future lectures. I think Luke's alluded to it in terms of how Cre works. But essentially, one thing you should also appreciate is they have 3 different retro capsids here that they've injected all into the same site. And you can see right away in the panel below that they all have different tropism, essentially. So even the ones that have been engineered, you can see that some of them are preferring to go to cortex, some of them to some of these amygdalar areas. This is like caudate, I think. So basically they have different places that they're going after being injected. So there's a lot of troubleshooting that might be necessary if you want to use viruses for retrograde or intrograde tracing. But they did compare it to some other AAVs, just normal serotypes that do have a little bit of tropism. I didn't have time to discuss canine adenovirus, but it was basically the gold standard other than Florigold for retrograde viral tracing. AAV-DJ looks like it does have some retrograde tracing. So if you are using that serotype, you might want to keep that in mind. But here you can see this huge improvement with AAV2 retro. But basically this was optimized for this particular projection from BPN to cortex. So that's probably why I can't get it to work, really. Yeah, well, and the fact that the Cre-dependent is much less successful. But we can talk about why that would be.
Anita?
Yes.
How did they, either in this case or in general, check the injection site with those retro AAV to be sure that they injected where they thought they were injecting?
For viruses, it's mostly done by needle track. I don't think they co-injected a dye, at least in this case. So you would probably just check for your needle track. In this case, I don't recall the specific methods, but most likely they just mixed them in the same pipette and they all got delivered to the same place. So they probably didn't go in 3 separate times with the different viruses. So what's shown here is probably all coming from the same pipette. So I think here they co-injected, yeah, it looks like they co-injected an EGFP. So they had the retro type with tomato, and here they had AAV1 with GFP. So then the GFP stays local, and you can see the retrograde transport of the tomato.
The R doesn't mean that it's also retro on the GFP?
Sorry, so our AAV is recombinant.
Oh, no, it's, oh, okay, my bad.
Yeah, yeah. File that under things that you don't know you need to know until you need to know it.
Okay, got it.
Yeah. So basically they used another clever method of basically using Cre-dependent recovery of AAV capsid sequence and essentially went after it the same way. They had capped proteins. They diversified a certain region of it. The peptide sequence specifically is listed in the paper, and they made it dependent on Cre recombinase. They had different Cre lines, and in this case, they were saying they specifically chose CAM kinase Cre as well as GFAP Cre and maybe GAD Cre, I forget. So they wanted to check for excitatory, inhibitory neurons as well as glia to see if they could get expression. In this case, PHP-EB. They're trying to inject something basically into the body and get uptake into the brain. And so basically with the wild type PHP-B, they got some uptake into the brain from a peripheral injection. And here they get much more enhanced signal. So here you can see in a section, they get much more enhanced signal using this. And more recently, they've shown that there is obviously a receptor, as I've been trying to emphasize throughout this talk, that there is a mechanism that allows this uptake. So basically, there are strains of mice, which is a little disturbing. So hopefully you don't work with any of these strains of mice if you're trying to get expression in the brain. So different strains of mice, basically they're inbred. And so basically they are inbred from a stock for like hundreds of years. And so they can have these certain SNPs that are characteristic of the strain. So all mice, for example, BALB-C has some pretty famous point mutations that make it, this is the albino one. So it changes the coat color. I think they're also blind or something weird like that. So you have to think about that when you're using inbred strains. So basically there is a receptor for PHPB as well as EB, it's the same receptor, Ly6A. And so there are point mutations in all of these strains of mice and they get almost no expression. So this could be a reason why you're not seeing uptake of the virus if you happen to be using one of these strains. Thankfully, C57 does use it. A lot of these are strains most of us are using, especially for transgenics. But just something to keep in mind that because these are receptor-mediated events, this is, if you're having issues and you need to troubleshoot, this could be part of that. Does anybody have any questions about AAVs before we move to rabies?
Could I make one comment about AAV, which is just to address a really common misconception that people have?
Yes.
And that's about titers and titering. I don't know if you're going to talk about that at all, but people typically, so you can do things like qPCR titering, where you're just using qPCR to count the number of viral genomes, like the number of DNA molecules in the tube. And then you can do what's called functional titering, where you actually, for example, like infect some sort of cells and then keep diluting it and find out what's the smallest dilution you can use that actually infects the cells. So almost nobody does functional titering. You do qPCR titering. So, but the thing is, if you boil a tube of virus and kill it, its qPCR titer doesn't change. So it actually doesn't tell you, it just tells you sort of how many virus particles are in there. It doesn't tell you whether they're functional or not. So
in one case you have particles, in the other case you actually have infectious.
Right, exactly. And so one of the problems is that there's no way, there's no way to, for example, so a lot of these studies that say, oh, AAV9 is better than AAV2 or whatever. The problem is there's no way to compare a functional titer between two different serotypes. So it's like if you had a good batch of AAV9 and a bad batch of AAV2, and then you normalize so the qPCR titers are the same, then like, oh, AAV9 works much better in stridum. But the thing is, that's not, there's no way to really say that conclusively. That's just something to be aware of. You see these things and people have this false sense of rigor when they're using, qPCR titers that are like, oh, like 10 to the 11th viral genomes, but it's like, that's, a tube that's 1 year old versus a brand new tube with the same number of viral genomes are going to be different. So it's just something to keep in mind that we don't have a good solution for that.
Luke, are you saying that it would be good to do the functional titering with any batch that you get, or?
Yeah,
no?
So I came up with an idea for how to do functional titering and I was too lazy to implement it. But the problem with functional titering, the problem with functional titering is that you can't, you can only compare within a serotype. Like you could say, oh, this batch of AV2 was better than the last batch of AV2 or worse than the last batch of AV2, but you can't compare it to the batch of AV5. So the way that you would do functional titering, for example, would be to infect 293 cells and then use qPCR to look for second strand synthesis, something like that.
How about competition between AAVs? If typically the example you gave with the retro, the paper with all of the AAV retro, what do we know about that? Is it possible to predict if there will be competition between two AAVs, or when we have to inject two at the same site?
So I don't think there's competition between the two. What happens though, I don't know, Anita, if I want to like hijack this and whatever, should we? So
Go.
AAV, AAV persists epizomally. So the AAV genome, it forms these head to tail concatemers. And so what happens in the beginning, there's lots of monomers, and then there's recombination between the ITRs, and it forms these, typically they're circular tetramers, or what becomes the stable long-term form. And so what happens if you inject two different AAVs, especially with different promoters, is that they form essentially heterotetramers, and then you get interaction between the regulatory elements on the different AAVs. And so this is another unsolved problem. So if you inject like a CAMK2 promoter one with a KAG promoter one, then all of a sudden your CAMK2 promoter one's not going to be specific for, you know, projection neurons or whatever, because it's going to be in the same tetramer as the one with the KAG promoter. So there's all sorts of weird stuff like that. And then there's some effects that aren't understood.
If we have to inject two AAVs, it's better to try to have the same promoter.
I guess what I'm saying is that if you have two AAVs with different, if you have two AAVs with different promoters, the promoters will interact in such a way so that you can't trust that the promoters will act the same way as if they're alone.
Okay, got it.
Yeah, as far as I know, people can easily get expression of two things. So for example, if you're trying to use AAV and you have a low payload, you can just mix AAVs and get two AAVs expressed in the cells. So that definitely can happen. I don't know more than two, how successful it is. It may not be as efficient as they're gonna have potentially non-overlapping efficiency. So you're not gonna get 100% of cells with both constructs. But for sure, people use that as a strategy to basically get around the payload limitations for AAVs. So it is something people do.
So one quick other warning, I'm sorry to interject this. So one thing that is relevant is if you have a genomic Cre-dependent locus and you inject a Cre-dependent AAV, it will inhibit expression of that locus. So if you have, for example, a Cre-dependent channel rhodopsin mouse, and then you inject in like a Cre-dependent GFP virus, it will inhibit expression of the genomic channel rhodopsin. If the lock sites in the AAV backbone are compatible with the lock sites in the genome, and that's been shown many times. So that's something that's a mistake people make a lot.
Yeah. Yeah, if you can, I actually am not familiar with that stuff. If you want to send me that, I can include that with the conditional expression stuff for the next lecture.
Sorry, yep.
Okay, so I just have a couple more viruses. I won't go into as much detail. I just wanted to give some insights with AAVs because I know a lot of people are using them and they're most of what's commercially available right now. I did want to talk a little bit about rabies virus and how we use them because I think there's a lot of confusion in terms of how we're using them in the lab because obviously they are not wild type. We cannot use wild type rabies. We would definitely be at risk because they do have tropism in humans. So basically there are some essential components of the rabies virus that are of interest for neuroscience, and that's the envelope protein, the glycoprotein, and then of course the genetic material. We're going to completely take out all the virulence factors and replace it with our genes of interest. So with wild type rabies, basically they get into a cell. It's actually thought to be mediated through nicotinic acetylcholine receptors, which I think is kind of interesting. So that allows it to go into a cell, it amplifies, and then it's coated with rabies glycoprotein. Rabies glycoprotein interacts with a receptor on the next cell and then can be taken up. And this is why when it buds off of a presynaptic side, it can infect the postsynaptic side and be transported. And that's how it's crossing synapses. So obviously, this is why rabies is so deadly. If you get bitten or something, it's going to start spreading through your entire nervous system. Obviously, why we do not use wild type in the lab. So we're not using anything like this, but we are going to take advantage of this glycoprotein and envelope protein to get expression in animals. So basically, back in 2010, they engineered this way, again, as I've talked about before, of taking out all of the virulence factors and then basically taking the G protein out of rabies. So basically here is what is the rabies. So rabies itself is not replicative. All it's expressing is your gene of interest, more or less. So in this case, M. cherry. And they've taken out the G, and the G is now getting expressed. In this case, it's expressed in trans, but here what in trans means is that we are injecting separate AAVs containing the rabies G protein and then an avian envelope protein. So basically the two ways that we're now protecting ourselves is that we've taken out both the envelope and the G protein. So this is called G deleted rabies, and it's pseudotyped with envelope A. So envelope A is avian, so presumably it would not have human tropism. And then we can express those in trans through a separate AAV infection. And then this actually is done in a separate infection. So basically you deliver the AAVs that contain your avian receptor as well as, your avian envelope receptor. So now this rabies is pseudotyped with avian envelope protein. You have a specific TVA receptor. And this is expressed Cre-dependently, so it's in specific cells. As far as I know, there's not a non-Cre-dependent way to do this. I don't think you would want to, you get too much expression. And then the G protein is delivered separately here. So it's not inside this rabies protein. So it has to get the G protein only in the cells expressing Cre. So then once you have this Cre-dependent expression of TVA and G protein, then you go back with your basically gutted rabies virus. The only thing it has is this ENV-A expressed on the rabies virus, and then inside of it is M. cherry. So it can basically now, because these cells are Cre-dependently expressing TVA, and it has the pseudotyped capsid for A, now it can infect those cells. The G protein is here too. The G protein will coat the capsid and allow it to go one time. But now in this cell, which gets infected, this cell does not have the G protein because that was expressed through an AAV, and it does not have the avian receptor, so it's gonna stop here. It cannot continue to cross synapses because we've taken out those factors. They're only expressed in this primary neuron here. Does that make sense to everybody? So on the next slide, I'll show, this is the actual injection strategy. So first, and in this case, this was using two separate AAVs from this paper from 2012. You basically go in with your TVA and your RG, you co-infect them in a single pipette, but you're mixing two different, in this case, it was AAV5 and AAV8. You inject those together. Some percentage of cells are going to take up both of them. And so that means that the virus, the rabies that you come in with, so this is again, pseudotyped with the avian receptor, and it's G deleted because we put the avian receptor here. So this is avian pseudotyped for the envelope, and then the delta G. So I'm saying this a lot because I feel like people get very confused. So you have all of these things are expressed in trans. The only thing that rabies has is this avian envelope and the GFP. So then in this case, your starter cells, you're going to be able to identify them because they're going to be both red and green. And then the presynaptic cells are only going to be green. And again, it's only going to cross one synapse because we only have expression of the RG and TVA in the starter population. And so in this case, they were comparing, I think this was a TH Cre mouse they injected into the starters. And again, the rabies and the starter viruses, they go into the same location. So you're not changing where you're putting your injection pipette. You want to come back to these starter cells for all three viruses. So then you can see a different pattern if you have TH cells that were infected from the VTA versus the substantia nigra. So you can see the presynaptic labeling across the brain. So this is handy because it's monosynaptic. It tells you the first order connectivity in a retrograde manner. So the inputs to your neurons of interest. And these were originally developed The System, again, by Ed Callaway, and a lot of these viruses are available at the Salk vector core. And you can, I think this is my last slide on rabies, but I'll just mention, in this case, they're just using a reporter, but you can also deliver other payloads now, such as channel rhodopsin, GCAMP. You could put CRE and do a second order tracing from those cells. And I'm feeling very emotional because I had ordered the stupid self-inactivating rabies. It turned out there was a point mutation and it wasn't actually self-inactivating. So anyway, anytime you're ordering viruses, they're all, as I showed you, they're all being kept in cultures. So they're in cells when they're being made. So if the cells get any kind of contamination, that can propagate to your rabies. So anyway, there's supposedly self-inactivating rabies that should be long-lasting and stable. So there is a kind of a 7-day limit, basically, on how long between the infection with rabies and when you want to observe is 5 to 7 days, because after that, you're actually going to get a decrease in the cells that you have labeled because they start dying. So these were supposed to inactivate the rabies such that you got expression of your payload and then the rabies stopped expressing. So, but it didn't work. So I'm feeling very sad about that. And I'm also sad because the vector core didn't actually tell me. I learned it from someone else. So a few $100s down the drain. All right, pseudo rabies, totally different. I don't know why it's called rabies. It's completely not rabies, okay? It infects pigs, it's different, and you know, it's not just coming from me, okay? If you go to Wikipedia, they're gonna tell you, don't confuse this with rabies, okay? I don't know how many times I have to say it, but even Wikipedia knows. Rabies and pseudorabies are not the same. They have different biosafety level, they have different everything. So it's just, so pseudorabies is the actual virus. In the case of pseudotyped rabies, it's rabies that we engineered. Okay, so that's the difference. It's a coincidence that pseudotyping and pseudorabies sound the same. And I don't know how many letters I had to send to the Animal Institute about how this was not pseudorabies and pseudotyped rabies were different, and that we were not going to get rabies. I don't know why I had to explain this, but you're not going to get rabies from G-deleted rabies because there's no infectious material inside of the rabies. Anyway, it's giving me a lot of headaches, but it's not pseudorabies. Pseudorabies is really tricky to work with. It's very toxic to cells. There are engineered ones. BARTHA is what BA stands for. That's the person that engineered the pseudorabies viruses for tracing. So I just wanted to highlight one case where they've used tracing. In the literature, I haven't seen pseudorabies recently. It's pretty good if you want to do like developmental tracing in young animals. It's not so bad. You kill them in a couple days anyway. But if you're using them in adult mice, they can die very fast and you're not going to get expression long term. But basically, I just wanted to show that for these different constructs of pseudorabies virus, that basically it's crossing synapses. It keeps crossing synapses. So in the case of pseudotyped rabies, we have a monosynaptic only. In this case, it will continue to cross synapses, and it's basically based on time. So how many days after infection that you sacrifice the animal, you're going to get more. But then I want you to notice that you start getting less also after days because the neurons start to die. So you only have a few days that you're going to be able to use this. But you can, if you infect the VNO versus the olfactory epithelium, you can get differential trafficking of the payload for the PRV virus. So I think these are like BSL-3 or something. There's someone in Catherine's lab now doing it, but he's at the Salk. I don't think there was one BSL-3 available for Catherine's lab to use. So just for your information, pseudorabies does not equal pseudotyped rabies. Does everybody understand pseudotyping generally and what a serotype is? Just, okay. Just make sure you learn one thing at least from this lecture. Okay, and then I'm just going to talk briefly. I didn't have time to get into detail on how herpes simplex viruses were trafficked and taken up and expressed and all of that stuff, but essentially it travels anterogradely, and this has been known for probably 15 or 20 years, and actually Gary Schwartz in the Department of Medicine at Einstein has a paper where they injected an HRP, I think, in an HSV 129 vector. So it's not actually HSV. HSVs can be used just for infection of cells, but the 129 version, which I think was isolated from a human patient that had like severe encephalopathy with HSV. So that's how they identified it. Basically this HRP delivered with a HSV H129 was delivered to the stomach, and then they could see labeling in the paraventricular nucleus. More recently, David Anderson's lab has made a delta TK-TT, so basically removing some of the toxicity, because this is a highly toxic virus. It causes a lot of immune response, very short-term staining. In this case, he's showing expression from this mutated version of H129, showing expression from cerebellum to some of the disynaptic connections using this promoter. And I've seen a couple of uses of the H129 delta TK. For example, Tali Kimji had a paper in 2014, and I just saw a paper from 2019 doing something with olfaction. But I don't know of widespread use of this, and I think it's because it's still pretty toxic. I think that it's maybe there aren't a lot of cells by the time you kill your animal and want to look. And again, this would be timing based, so you might only, I think the most people have done is disynaptic, so crossing 1 or 2 synapses. So in the case of the Scott paper from 2014, I think they only looked at the first order connection anterograde. And basically they wanted expression of the fluorophore in this cell. So basically with normal projection tracing, you just have the projections, but you don't know the identity of the postsynaptic cell. In this case, the postsynaptic cell would express GFP, so you could, for example, sequence it or counterstain it with other markers. I don't think this is widely used, so that's why I didn't spend a lot of time on it. I'm assuming that there's probably further development being done on the HSVs to make them less toxic because they do travel anterogradely. So just to wrap up, there's some good resources out there for viral repositories. Back in the day, you had to make viruses or be really good friends with somebody that made viruses. Now a lot of stuff is commercially available. If Adgene doesn't have the virus already packaged, they may have the plasmid, and so you can do the cloning yourself or potentially have them packaged into the viral serotype of your choice. Again, they're supplied in trans, so I'm assuming they have some sort of good platform to just add whatever capsid protein you want to the gene of interest. And it has really good annotations, and they have a blog that a lot of the schematics that I showed are from the Adgene blog, so they have a lot of good information if you're just looking for notes on the plasmids or general methods. The UNC Vector Core, they mostly have DREAD constructs, and then some other, there's like a few investigators that deposit their stuff here. They had a big contamination problem in 2014, and I never went back, so I don't know what the status of UNC Vector Core is now. I've already given you my sob story about Salk, but I'm sure some of their viruses are fine. They have some other cool tools that I don't have time to talk about during this lecture, but if there's something in particular you want me to cover, we are going to cover cre-dependent mechanisms in another lecture. So if you have any particular requests, so we're going to talk about, using transgenes, knockouts, knock-ins. We're going to talk about CRISPR, sequencing, pharmacology and lesions are still on the list of things to cover. So if you have particular topics you want me to discuss, I'm happy to add them in, since we're in this online format, and I haven't actually done the lecture for the next time, so still time to put in your requests. So any other questions?
Anita, I was just wondering if you were going to go over the trio method at all.
I didn't have time in this method. That can be, I can cover that in the CRE-dependent, because it's a little bit feeds into the combining some of these. So this is like really to give you an idea of like how the viruses are working, and then we can talk about how you can combine some of these technologies to get some cute things. So we can talk about C-trio. Anybody have other requests? They have some really cool stuff in fly that I'm not sure if it works yet in mammals. The grasp and tango and some other acronym-y stuff.
And how about light sheet microscopy and what's the name? Clearing methods. You covered it a little bit today. Is that going to be it, or?
I wasn't planning to go through that, but if you guys are interested, I can give some overview of some of the clearing stuff.
Depending on if it's likely to come to the department or not, which I'm not sure. But
I would like it too. I'm planning.
I would like it to too. So yeah.
All right, and we will discuss, we'll give you an update on how we're gonna go through the final presentations, because most likely we'll have to pair people up at least, if not more, because we only have a couple hours to have you guys present, but more or less it will be presenting what techniques you would apply to a particular problem and what maybe the advantages and disadvantages would be of using that approach. So just a little mini presentation, and we'll figure out how many of you will do it together and how much time you have to present by next time.